Hello everyone, I am Laura Shiromi David from Monogastric Research Centre in School of Agriculture and Environment of Massey University, New Zealand. My presentation topic for APSS 2021 is True Ileal Calcium Digestibility in Soybean Meal and Canola Meal without and with microbial phytase for broiler starter and finisher. Let's have a look at introduction first. Currently, there is a move towards the use of digestible phosphorus in diet formulations for poultry due to phosphorus excretion into the environment and increasing prices of inorganic phosphates. However, total calcium is still considered in diet formulations due to lack of data on digestible calcium content in feed ingredients. As we know, phosphorus and calcium are closely related in their metabolism. High dietary calcium concentrations are known to reduce phosphorus availability in poultry. Therefore, measurement of calcium digestibility in feed ingredients is important to formulate the poultry diet with proper calcium and phosphorus ratio. Canola meal and soybean meal are two plant-based protein sources which contain about 6.8 and 2.9 gram per kilogram calcium respectively. Data on ileal calcium digestibility of calcium sources are now becoming available. Our previous studies reported the ileal calcium digestibility in major inorganic calcium sources and animal-based calcium sources. However, digestibility studies on plant-based calcium sources are limited. To the best of our knowledge, currently no published data available for the calcium digestibility in soybean meal and limited studies are available for the calcium digestibility in canola meal. As you can see here, only 2 studies reported very low values of calcium digestibility for canola meal, which were based on purified diet. Microbial phytases are now routinely added in poultry diets to improve the bioavailability and absorption of phosphorus and other minerals. In general, the phytase dose recommended for broilers is 500 units per kilogram. However, higher doses over 4 times the recommended dose and referred to as superdoses are currently being used by the industry and reported to improve the growth performance and nutrient digestibility. The effect of phytase on calcium digestibility, however, is contradictory. Some reports indicate that phytase addition improves calcium digestibility, whereas others have found little or no benefit. Therefore, the primary objective of the current study is to determine the true ileal calcium digestibility in soybean meal and canola meal without and with microbial phytase during broiler starter and finisher periods. Secondary objective is to investigate the influence of microbial phytase dose on true ileal calcium digestibility in soybean meal and canola meal. Moving on to materials and methods, 6 experimental diets were generated using 2 ingredients, soybean meal and canola meal, with 3 phytase doses, 0, 500 and 2000 units per kilogram. The experimental diets were maize-based and the contribution of calcium from maize was assumed to be negligible. A calcium and phosphorus-free diet with no added phytase was also developed to determine the endogenous calcium losses. This is the ingredient composition of experimental diets. Here, soybean meal and canola meal were included at 50 and 30% respectively in respective diets and served as the sole source of calcium. Calcium and phosphorus-free diet was based on maize starch and dextrose. Experimental birds. There were 2 experiments, and the experiment 1 was conducted with broiler starter and the diets were fed for 3 days from day 19 to 21. Similarly, experiment 2 was conducted with broiler finisher and the diets were fed from day 40 to 42. The dietary treatments were replicated 6 times and each replicate had 8 male Ross broilers. Let's see the results on true ileal calcium digestibility. There was an ingredient phytase interaction observed in broiler starter and finisher. True ileal calcium digestibility coefficients of soybean meal and canola meal without added phytase were determined to be 51 and 53% respectively in broiler starter and with low values of 33 and 22% respectively in broiler finisher. Increasing phytase dose increased the true ileal calcium digestibility of canola meal in both starter and finisher, but the calcium digestibility in soybean meal increased only at the superphytase dose in finisher. The phytase response was more pronounced in canola meal than in soybean meal, and a possible reason for this difference could be the higher phytate concentration in the canola meal. Regardless of ingredients, the superphytase increased the calcium digestibility as twice as the normal phytase dose. The difference in calcium digestibility values between starter and finisher, regardless of ingredient, clearly indicates the influence of broiler age, which confirms our previous finding that the calcium digestibility decreases with advancing broiler age. Let's see the results on apparent total tract calcium retention. In broiler starters, only the main effects were significant. The calcium retention was higher in canola meal than in soybean meal, and this could be due to a relative higher calcium concentration in canola meal. And the calcium retention was increased with phytase doses in both ingredients. However, in broiler finisher, there was an ingredient phytase interaction where the retention was increased by both phytase doses in canola meal, but only by the superphytase dose in soybean meal. Conclusions. The true ileal calcium digestibility coefficients of soybean meal and canola meal for broilers were determined, which is the first report for soybean meal. Second, broiler age influences the digestibility and retention of calcium in soybean meal and canola meal. Third, supplemental phytase is consequential for calcium absorption and retention. Especially, the superphytase dose improves the calcium digestibility as twice as the normal phytase dose. Thank you very much for your kind attention.